Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Jean-Marc Cappia and Armin Hauk from Sartorius Stedim Biotech.
Okay, so thank you for the introduction. So the context for this presentation is the increasing adoption of single-use system is raising a growing concern for extractable, leachable, and particles. And then the goal for this presentation will be to show you how applying good science and stringent process control will provide well-characterized and reproducible extractable, leachable, and particles, which in turn will give consistent biocompatibility of single-use material and components with biologicals. So single-use systems, together with process intensification, have been the key tools used by the industry and the biotech industry today to improve production efficiency and to reduce the cost. So there's no need to repeat all the benefits of single use, but the key point here is that we will expect yet a massive growth of the single use, especially when you see this chart here, where only 20% of the commercial process steps today are using single use, and we have this clinical pipeline with 80% penetration of single use. So every time a new biologic switch from clinical to commercial, there is a huge increase of single use in commercial processes, and of course this is due to the benefits, but we will see also that it brings a certain challenge. So the interesting point anyway is that the view of the benefit of single use is accepted and acknowledged by both the end users and the authorities. And here I'm quoting Patricia Hauk from the FDA, and that was during one of the FDA ISM international meetings. So she does recognize all the benefit of single use in terms of safety, in terms of economic, in terms of providing more flexible processes. But on the other hand, she raises these key challenges. So you see that together with integrity and supply chain and change identification issues, the biocompatibility of single use with biologics is at the center of the regulatory authority attention. And behind biocompatibility, we have three topics, which are the extractables, the leachables, and the particles. So what are we doing with this? And here you see a graph with the extractables, and you see that extractables will be generated by the materials, so the different resins used for making the films, the bags, the tubings, the connectors, and the filters, but also the process. When you extrude, when you weld, when you sterilize, you involve high temperature, you involve gamma radiation. So all this will generate extractables. So that's why, first, you need to know your material and your process. You want to fully characterize your extractables. You need to know the composition of your resins, the different additives, down to the cast numbers for getting to this characterization. Second, you need to control your process. If you do not control your resin, your extrusion temperature, your sterilization time, etc., you may have a different profile of extractables. So then, when you know and control your process, you can know exactly what extractable level you will obtain, and you can ensure their consistency. Which, by the way, facilitates the validation by the end user. So we'll have to correlate these extractables to its own process conditions. So in this slide, what we have done is we have first adopted our internal extractable approach. And here we are using very aggressive conditions with 100% ethanol, pure water, 1M and sodium oxide, and 1M chloride acid, so, and the multiple time points, and we do that for all the different components. So here the goal is to really characterize all our materials, components, filters, tubing, connectors, etc., to make sure that we understand what potential extractables could be released into a biotech process. So that was done on more than 300 components, and we have created here a huge database of different extractables with multiple type of analysis like HPLC, gas chromatography, etc., to make sure that we have a full screening of volatile, semi-volatile, and non-volatile extractables. So that's the first step here. The second step we undertook is applying the BPOG extractable. And here the goal is to get extraction conditions and extraction solvents that will be closer to actual normal process conditions. And you see here that the pH and acid conditions are milder to meet the process conditions. And there are also other solvents like the twin or the salt and others like phosphoric acid that are here to mimic the type of solvents used in the industry. So again, here a huge database of extractables data have been created. And now let's see what use we can make of these two sets of data. So first, you can use the so-called in-house Sartorius extractable data and compile them into an extractable guide. And here the main challenge will be to move from component data that we have for every tubing, every connector, etc., into full package of extractables that will be, let's say, representative and relevant to a complete single-use assembly. Because end users are not just using a tube or a connector or a filter, they use an assembly made of these different components. So that's the first, let's say, document that we can generate with this data. In second, we can also now relate our extractable data, be they the one from our internal Sartorius approach or the one from the BPOG, and relate them to actual process conditions. And the idea here is to kind of create digital extractable data for a lot of applications, mainly in downstream. Normally, we do not need to evaluate actual leachable, for which here you need to really evaluate leachable using the normal process conditions and the drug product. In this case, you can only use existing data and relate them to the end user process and create so-called digital extractable reports. And we will give more details on this, especially with Armin in the rest of the presentation. So the first challenge here is now we get all this extractable data for the different components. And we can create extractable guide for a tube, a filter, a bag, etc. But now the question is, what do we do when we assemble all these components into a single-use solution, which is what is used by customers. And here you see that you have to combine a lot of components, a lot of materials. The complexity also comes from the different surfaces and lengths of tubing, for instance, that are used. So there is a kind of intuitive approach to scale by surface and then apply a cumulative approach. But this is in conflict with physical principles. It has been laid out also by Dennis Jenke, who is an expert in extractables, but Armin will also explain this in more detail. In fact, extraction is most of the time either a diffusion or equilibrium control. So most of the time, when you have short contact, the extraction will be more governed by diffusion. So here, a surface scaling will make sense. But for long contact time, you will reach an equilibrium, and here a partitioning model for scaling should be better. And you can see here an illustration of what I'm saying. So you see that not only depends on the time, but it will depend on this coefficient of partition for the different extractables. So you see how complex it is, because different extractables from the same material will behave differently. And you see here that for this first, where the coefficient of partition is small, then you don't reach the equilibrium. So you could scale depending on the size or depending on the number of process steps. But when you go to high coefficient of partition here, you see that if you extrapolate based on the scale, based on the number of process steps, you will make a big error, because here you have reached the equilibrium, and it doesn't matter whether you use 1, 2, or 3 bags in series or only one. So that's a key observation that Armin will describe in more details and more scientific data. Okay, so with this, then we have to work and develop the software, because as you can imagine, this calculation of extractables is very complex. So today we have all the data available for the different components. So, and we are now developing a software able to select the different components when it comes to a full assembly, and then make the calculation of the potential extractables specific to the assembly. So the second challenge and work that the software will have to do is now to also connect this extractable data with your process conditions. And here again, the system will look at the different components, A, B, C, with the different type of extractables. We'll also look at the solution that is used in the process, but also the process conditions like contact time, temperature, etc. And then we calculate extractables. And the goal here will be that the system generates extractable data for complete assembly in the first step, and then extractable data that can be related to your process and that can be used as a potential leachable simulation. So I'm putting here a picture of the FDA with Armin and other subject matter experts visiting them to tell you that we have also presented this approach to the FDA, and that we had a very good feedback, and this approach is well recognized, which will allow the industry to save time and money on all the extractable evaluation, especially in the area of downstream processing. Of course, we are not saying that for the final product this will replace actual leachable studies, but a large proportion of the work done in downstream processing could be achieved using this digitalized extractable process. Okay, so that's it for the extractable. And the other piece that is critical when it comes to single use is the particles, because for the value of single use is that it's ready to use, and then you do not have to clean this equipment before you use them. So, but on the other hand, when single-use products are manufactured, even though we manufacture in the ISO 7 area, they are containing visible particles. So the sub-visible particle has been under control for years, and this is not the issue. Now the new challenge is controlling visible particles. And here you see that we have put in place a system of monitoring and tracking particles in the last 1-2 years, and you see here the trend. So we have also compared the level of visible particle contamination of our bags together with other competitive technology to check that all suppliers were in the same high level of quality. So you see that at the end, yes, there are low level of particles, but yet we cannot claim that there is zero visible particles. And this is what we would like to reach in the future and meet the famous statement of the USP 790 that we should have essentially free of particle products. But the key point here is that at the end, we are not at this zero particle, but the key point is that by installing such a control system into our quality control now, we have a good identification of the type of particles. So then we understand the origins of the particles, and by making this measurement, we are and putting ourselves a limit and specifications, then we are forced ourselves to understand the root cause and to correct. So by this, we will create a continuous improvement and hopefully reach the zero or essentially free of particles in the near future. So that's the context for extractable and particle. And I would like to hand over to Armin to get to the technical details now.
Okay, I'm Armin Hauk, and with the following slides, I will briefly summarize our Sartorius research initiatives. The aim of this initiative was to develop methodologies to use extractables data in a much more thorough way, and to go beyond this approach to just simply measure extractables. The thing is that we can look back on more than 20 years of experience in the extractables and leachables area, and we saw that at a certain point, if we would combine our empirical knowledge with physics, with chemistry, with material science, with mathematics, then we can come to from this more empirical point of view, to an understanding of extractables. That means, first of all, we need to develop an extractables method which fits our purpose. And with this extractables method, we try to get comprehensive extractables profiles of our devices. And combining this knowledge, we would be able to go ahead in a step number 2, where we try to find methodologies to calculate extractables for materials, devices of different sizes, and device combinations. These are assemblies. And in a third step, we also want to use mechanistic modeling to predict leachables in whole processes. But this requires, as I said, that we start with our empirical knowledge, combine that with good science to develop a method, to develop a comprehensive extractables profile for our devices, and then to go ahead with the other steps. As I said, the first step was to develop a suitable analytical approach. We have this approach. This is our Sartorius extractables approach. This was developed in a greenfield project, and greenfield means here really we started from the scratch. I developed a new method. It is based on a reduced set of extraction solvent, but state-of-the-art analytics. It requires no sample prep. It reduces the risk of analytical artifacts. It enables a straightforward identification of extractables, and it allows scaling and combination of data, and it allows also customers' risk assessment based on identified and quantified compounds. And last but not least, the justification and the description of the method was published last year in a paper from BPI. This method enables us now to analyze our devices. And I can say that that we are proud, more or less proud, to say that today we have the lowest number of unknowns compared to any other methodology which is on the market. So the lowest number of unknowns in our devices. Today we have nearly identified 95% of all the compounds which may occur from our devices. For the majority of the extractables, we know the origin. And what was, of course, encouraging us was the quantitative extractables profile is under all circumstances a function of the amount of extractables in the polymer, the extraction time, the extraction solvent, and the temperature. And this again leads us to the situation that it became clear it is necessary to bring physics, chemistry, and material science in the center of the discussion to go ahead. If you want to use scientific models, or if you want to use physics, then of course you need a model you need a description or a model for your extraction. And extraction is commonly a situation where you have a plastic phase which is in contact with a liquid phase. There is a compound in the plastic phase which is migrating in the liquid phase. The driving force behind is the disequilibrium between the two phases. While this process happens, you have a migration of the compound to the liquid phase, and this process stops when the two phases are in equilibrium. Such a situation can be described with a so-called migration model. And here you have a cross-section through these two boxes. On this side, you have the plastic phase with the well-developed diffusion gradient of the extractables, the concentration jump at the interface, and the concentration in the liquid phase. Such a system could perfectly be described by fixed second law of diffusion if you are not in equilibrium, and with equilibrium equations if you are in equilibrium. And exactly this approach was chosen to determine our extractables, to analyze our extractables, and to go ahead in our approach. Because this physical description has certain consequences, direct consequences. For example, as Jean-Marc mentioned before, you cannot scale equilibrium data, for example, just by surface area, or can add them. But you have the probability that you say, okay, if I have extractable experiments with short contact times, then I can, of course, assume a diffusion-controlled extraction, and then scaling via contact surface area and a cumulative approach is possible. If we have longer extraction times, then of course you have to consider that you are running into equilibrium, and then scaling with a partitioning model is required. We have developed both algorithm for scaling for short contact times and an algorithm for the calculation of extractables under equilibrium. And these algorithms are used in the before-mentioned database to calculate extractables, to scale extractables for devices of different sizes, and to combine extractables for assemblies, for example. But you can even go one step ahead. Now let's assume you want in a third step also analyze what's going on with extractables in the situation that they became leachables. That means in a process. Then of course you have to consider that the common perception today is that single-use systems are sources of leachables. And the extrapolation from leachables data will lead to the situation that everybody considers that there should be a high load of leachables in processes and in products. But if you are analyzing products in a process qualification study, for example, then you will not find leachables. You have used thousands of plastics, but you will not find a lot of leachables in your product. And there are also publications out, for example, from Nicole Scherer. She showed that in a downstream processing, the load of leachables is going down during the processing. Here you have the example from a fill and finish line, where she could show from thawing the product, filtering the product, transferring the product to the filling line, you have a decreasing load of leachables. And there is another presentation which was given by Jessica Shea some years ago where she could show that in a model process you have rising concentrations of leachables during the time in the bioreactor, but as soon as you start with harvesting, the leachables are falling down to a very low level. They are coming a little bit, they are going a little bit, and in the end of the product there is nearly no leachables. What is the reason for that? The reason for that is there are obviously sinks of leachables, very, very efficient sinks of leachables in the processes. And if you want to make a modeling of a process, then you have to take into account the sources, yes, the distribution, and also the sinks of leachables. And this is the so-called fate of leachables modeling. This fate of leachables modeling is using the so-called dynamic box model. These are mathematical models where you break down your processes in different compartments, and you calculate in each of these compartments the sources, the sinks, dilution, and partitioning of certain compounds. And you can do that with leachables in the flow of a process. And I will show you now results of a very, very simple model where we have 7 different compartments modeled, where we, in all these compartments, modeled the surplus of leachables which coming into the process step by migration out of the materials, the distribution in the compartment, and the removal in the compartment. Here we have considered a bioreactor, a harvest step, a handling after harvest, different filtration step, a ultrafiltration, deafiltration step, a tangential flow step, and then a fill and finish line. In all these boxes I calculate, as I said, sources, sinks, and distribution of leachables. And the results you can get from such a model, and I say this is quite a simple model, you can of course make this model much more complicated, is seen on the next slide. This is the model results of a leachables like a deterged butylphenol, a very common extractable. And you can see the red bars are the surplus of leachables from the previous step. The green bar are the leachables increase in the step, in the compartment, and the blue bar are the removal of the leachables in the dedicated compartment. And you can see during the time in the bioreactor you have a rising concentration of leachables while you are exhaustively extract your bioreactor. The harvest step removes a lot of leachables because leachables are removed with the cell debris out of the system. The filtration steps are bringing some leachables and removing some leachables. Ultrafiltration, deafiltration will also remove some leachables, and in the end you have only a very, very low load of leachables. You can make a calculation also for other compounds. This is a compound like a caprolactam. Again, you have a rising concentration of caprolactam during the time in the bioreactor. Caprolactam is not as well adsorbed on the cell debris and removed with the cell debris, but caprolactam is, as a good soluble compound, efficiently removed via the tangential flow step, the ultrafiltration, deafiltration step, and again, you will end up with very low amount of leachables in your product. So this fate of leachables model, in its very, very simple way, can already now much more and better and quantitatively predict the load of leachables than any other worst-case model which is used so far. and this model supports quantitatively and perfectly the proximity to patient concept. So this model will be further developed by us, and then we will use this model in a modular way to calculate leachables in downstream processing. With this slide, I'm at the end of my talk, and I'm open for questions. Thank you very much.
Okay, great. So it looks like we have a few questions, and one of them asks, which particle specification is claimed for the current bags when we order them?
What protocol is used for current bags if you order them? Of course, Sartorius will use our Sartorius Extractables approach to analyze the bags. As I said, the aim of this, the aim of the target or the goal, what we want to achieve is a full extractables profile. And this extractables profile can then be used in any other study, for example, in a simulation study or in a leachable study or whatever. But definitely, we will provide extractables data based on our Sartorius approach. And again, the big advantage of this approach is that you will get a full extractables profile with nearly no or only very, very few unknown compounds.
Okay,
Jean-Marc,
so
did you have anything to add?
I'd like to add, this is for extractables. When it comes to visible particles, in fact, we don't have yet a specification. So we have to be careful that we are talking about visible, because for subvisible particles, so the USP 788, which is subvisible, here we have the extraction protocol and the specification from the USP 788, and we claim that in our certificate of quality for the products. When it comes to visible particles, here we use the 790, and here there is no spec, as I said, and the statement is it should be essentially free of particles. So, and we know that this is not, let's say, possible to obtain in the short term, and this is why first we establish this monitoring, and monitoring is here to understand the root cause and to correct, and then reach in the future this essentially free. And then we also put in our process visible inspection, that we discard bags that will be inspected for particles, but just like the industry is doing on the final vials.
Okay, great. So does this approach apply to your sterilizing filters or only to bags?
No, of course we are applying our method to bags and to filters as well, also to tubings and connectors and all the other things. So we have a matrix where we define the different extraction conditions which are required for the different devices, and then we are testing all the devices according to our Sartorius approach. And I saw there is a question how we define short and long-term exposure. And the thing is, of course, for our devices we have for the devices which are used for long-term storage, we have kinetic data, so I know exactly when they are running into equilibrium, and I can say for typical bag materials, you can assume short contact times means time between some hours and days, and long contact times for bag materials and tubings, for example, means 70 days, 30 days, 70 days, then you are starting to running into equilibrium. And this is reflected, of course, in our matrix for testing.
Okay. Jean-Marc, did you have anything to add, or?
No, that's okay. Thank you.
Okay. We have a number of questions. So if we don't get to your questions, the questions will be passed to our speakers, but I'm just going to go ahead and ask the next one. What is the defined time frame for short-term and long-term exposures to be considered the diffusion and partition model?
Yeah, as I already said, for filters we use normally a short contact times as everybody else. USP 665 assumes short contact time. BioProcess requires short contact time. We also use short contact times, and for materials which are used for longer contact time, then of course we consider we have of course kinetic data. That means we know when these systems are running into equilibrium. And that means, for example, for a bag material, short contact time means some hours and days. and long contact time means, for example, 1 month. So this is approximately the time frame what we are talking about.
Okay, great. If the extractable data is available for all polymeric materials used in the process, can the fate of leachables model be used to avoid doing a process-specific leachable study?
I would say yes, this is the final goal. The final goal is that we would use extractables data and good physics and our modular system and calculate indeed what may come out of a process. I think that most probably authorities will then ask, please prove your modeling concept. That would mean that you have to make 1 or 2 distinct analytics, and then you can be sure that this model has made the right calculations. But this is right. In principle, you can then avoid a lot of process-specific leachable studies. This is true.
Okay. Well, if we didn't get to your question, again, they will be passed on to our speaker. So you can go ahead and type any final questions in now, and we'll pass those along. But thank you, Jean-Marc and Armin.
Thank you.
You're welcome.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing, and as a registered attendee, you'll receive an email with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Goodbye.
Bye-bye.